The history of medicine has been marked by improvements in diagnosis that have been important because they've changed treatment and improved outcome for patients. In cancer, our diagnoses today are done largely by looking through the microscope at the appearance of the cancer cells. Often what organ the cancer comes from is the most important way of making the diagnosis. But over the last couple of decades, we've begun to realize that a much better way to diagnose cancer is by the molecular abnormalities that distinguish it from the normal cells in the body. And these molecular abnormalities are often changes in the DNA sequence in the genome of the cancer cells. In a couple of large programs in the National Cancer Institute, we've been undertaking the sequencing of thousands of human cancer biopsies. One called the Cancer Genome Atlas, or TCGA, has been done in collaboration with the National Human Genome Research Institute. And the other is called Target, and this one focuses on pediatric cancers. So I'd like to give you a few examples from the recent discoveries in the TCGA program. The first is in lung squamous carcinoma. And here, molecular abnormalities were found in many enzymes called kinases that activate them and cause the malignant process. Fortunately, there are drugs already made that target these kinases. This has enabled a new clinical trial to be launched by the NCI called LungMap. And in this trial, patients with lung squamous carcinoma are being assigned to different treatment arms of the trial based on the molecular abnormalities of the tumor, one of the first times this has ever been conducted. And we aim to see whether that will improve the outcomes for these patients. Another notable success of the TCGA program was in gastric cancer, a particularly aggressive and lethal form of cancer. Here, the TCGA found that this one type of cancer may actually be 4 molecularly distinct diseases. What was fascinating is that one of these appeared to be driven by a virus, the Epstein-Barr virus, which is a very ubiquitous virus, but seemed to have caused gastric cancer in these patients. And in this subtype of Epstein-Barr virus form of stomach cancer, there are other molecular abnormalities that may give us clues to treating that type specifically. So we are hopeful that a comprehensive view of cancer derived in this way will improve our outcomes in cancer.